Thank you, and thanks to Leonard Sachs and his excellent team for hosting us here today. I also want to thank Brian for that perfect, super comprehensive setup presentation that he gave as ideal introduction to this panel discussion. I'm Jay Olson. I'm a biotech analyst at Oppenheimer, and my co-chair, Joel Sandler, and I are going to co-moderate this next panel discussion on Parkinson's disease and movement disorders. We'll focus mostly on Parkinson's disease. And, in the prep session, we spent some time talking about the enormous unmet need, which Brian so eloquently described. And I would just only add to that unfortunately, Parkinson's is becoming more prevalent, and it's not just as a result of the aging population. Age in itself is a risk factor for Parkinson's, but also environmental risk factors, microplastics and other pollutants have unfortunately increased the prevalence of Parkinson's disease. So it's an enormous unmet need. And as Brian indicated, there are a number of both disease-modifying and symptomatic treatments in development. And we obviously need both of them. And luckily, here we have on our panel several representatives of companies that are developing some of those treatments, including Gene from Gaia Therapeutics, working on an approach to GBA1 Parkinson's, and also Sam from Aspen Neuroscience, working on a cellular therapy approach to Parkinson's. So we're very lucky and grateful to have our distinguished panelists here with us today, and I want to thank them for joining us. And with that, I'll turn it over to my co-moderator, Joel.
Hey, everyone. Thanks so much. Thanks, Jay. Apologies for the little delay in the beginning, but we had some slides and that didn't work out. So we're just going to jump in because all you want to do is really hear from our panelists anyway. I'd like to just start by asking our panelists to briefly self-introduce and what their role is in terms of in terms of addressing Parkinson's. I'll just start very briefly. My name is Joel Sandler. I'm an associate partner with Ipsos Advisory Services. We provide strategic guidance for biopharma companies, large and small, particularly those that are looking to figure out how best to inflect value within the evolving competitive landscape. I'm a scientist by training, so I like to think about things in terms of what is the scientific rationale. I think represented across the panel, we can speak very credibly to the science and very quickly kind of ask the question about so what, how this translates. I would say we're in a moment right now, and I'd love to hear what people think, where we're transitioning potentially from symptomatic management of this disease to one that might be more disease modifying, and that is represented, I think, nicely by several of the panelists in terms of the mechanisms that are being developed here. I don't want to disparage or kind of put symptomatic management at a level down from disease modifying. I think that both played a critical role, and there are challenges in terms of demonstrating and validating modalities that can achieve disease modifying benefit. So we're going to keep the discussion today grounded in the science, but in a way that's relevant to the audience, talking about trial designs, biomarkers, maybe even taking a step back to start and asking about unmet medical needs and how we can learn potentially from Alzheimer's disease and take some of those learnings forward in Parkinson's and other areas of adjacency beyond Parkinson's into other movement disorders. But with that kind of rambling preamble, maybe we can just go down the line, Gaia, starting with you. If you can just take a couple of minutes, describe yourself and your organization and what you're doing with respect to Parkinson's.
Hello, everyone. My name is Gaia Skibinski. I'm a scientist at the Michael J. Fox Foundation. I predominantly work on the preclinical translational side of the organization. I don't really need to give that much of an introduction to the Fox Foundation because Brian just gave a 30-minute talk on it. But just to say, I'm here along with another colleague, Ryan, and we can assist you to discuss many different opportunities and partnerships that you can take part with. One is funding, but also access to resources that we have available, data and samples from patients from our longitudinal biomarker study, and then also being a part of our network. So we have thousands of people involved in our network. These are researchers, industry players, VCs, regulators who can help you and your programs on Parkinson's disease as well.
So I'm Gene Mack. I'm the Chief Executive Officer of Gaia Therapeutics. Characterize my job as basically staying out of everybody's way and clearing the path so that they can do their work. But Gaia Therapeutics is developing an allosteric modulator of glucose cerebrosidase, which is an enzyme that's prominent in the pathophysiology of Parkinson's disease, particularly among genetic mutation variant of that within the GBA1 gene mutation. So our molecule and its backups are meant to modulate the activity of GCase and improve, hopefully, the overall health and metabolism of the cell, and get to that biology and provide some sort of backbone of therapy so that those symptomatic therapies aren't really the only thing that patients can rely on.
Good morning, everyone. My name is Nick Manusos. I'm founding CEO of Kenai Therapeutics. We're an induced pluripotent stem-based stem cell company, or neuron replacement. We're off-the-shelf allogeneic cryopreserved version. We just entered the clinic. Our mission, if you will, is to restore what Parkinson's disease has stolen from patients. If you think about that from a disease modification standpoint, so we will first replace what Parkinson's disease has stolen, those lost cells. Second, our goal is to repair the circuitry, and finally to restore function and quality of life for Parkinson's patients. Again, we just entered the clinic. Thank you.
Sam Murphy, Head of Corporate Development at Aspen Neuroscience, also working on an iPSC-derived cell therapy for PD. We are in a phase 1/2A trial now, executing in moderate to advanced patients with Parkinson's disease. We announced last year some early safety and efficacy data from our first 3 patients. At 6 months, we will be reporting our top-line data later this year. And rising tides raise all boats for cell therapy. We're rooting for all teams. I think the point of differentiation for Aspen is that we are autologous, so we do not require immune suppression, which is an important factor for older patient populations. The first thing most people think of, you're autologous, how are you actually going to manufacture for patients and meet demand? So at Aspen, in addition to advancing our cell product, we are also pioneering a process which is modular to enable parallel processing for hundreds of patients. We have our own GMP manufacturing facility to support launch, and very excited for the potential, as Nick said, to restore function for patients.
Good morning, and thanks for the invite to be here. I'm Behrouz, one of the co-founders and CEO of Vincere Biosciences. I'm a neuroscientist by training, and I've been spending basically the past 20 years on Parkinson's disease, first trying to understand the biology that's underlying it, and then, of course, now translating that to disease-modifying therapies. We have small molecules that are targeting the enzyme USP30, and this target is really at the intersection of mitochondria and lysosomes, so it enhances and changes the threshold for mitophagy, which we know is a really important biological thing that is linked to Parkinson's disease. So we're hoping to slow or stop the progression, and we're getting into the clinic this year, so that transition from preclinical to the clinic is quite exciting for us.
Good morning, everybody. Thank you for having me. My name is Stefan König. I am the Chief Executive Officer of Merz Therapeutics. Merz is a mid-sized, family-owned private company focused on specialty neurology. And as part of that portfolio, we have a number of drugs actually that improve Parkinson's disease patients' lives more on the symptom side. And I was very happy to see from as the starting point of the Michael J. Fox Association to have these therapies stand next to each other. Now, I would like to see as many therapies from the disease-modifying end to succeed, but I think the pathway is quite long until we get there, and then not all of these therapies may actually have an effect on these patients. So symptomatic treatment will remain very important, and I'm very happy that Merz, as a mid-sized company, can address some of these markets. We've just acquired 18 months ago products from Therapeutics, with an important inhaled dopamine product that's treating the off symptoms that many patients incur. Many of these non-disease modifying drugs, they need mid-sized companies sort of safe haven for promoting them and for making them accessible around the world. And I think this is our mission at Merz Therapeutics.
Great, thank you for those introductions. For those of you who were here for the Alzheimer's panel, we heard about the analogy that was given from amyloid therapies to tau-based therapies and sort of trying to leverage some of the lessons learned there. We're going to extend that analogy from Alzheimer's to Parkinson's. And so in a disease where the underlying biological pathology has an onset several years before the presentation of clinical symptoms, we face a lot of the similar questions in Parkinson's as were discussed earlier in Alzheimer's, in terms of when is the right time to intervene, especially with regards to disease-modifying treatments, how to think about different endpoints, and a number of other issues that we could potentially borrow from Alzheimer's. So I'll ask our panelists to weigh in on that. Maybe Gaia or Stefan, if you'd like to start.
Yeah, I also just wanted to touch a little bit on the unmet needs. Stefan, you kind of brought this up again around the symptomatic treatments. I think it's really important, particularly for this room, to understand the value for us to go after symptomatic treatments, both motor symptoms and not the traditional canonical symptoms that people think about, but symptoms like freezing of gait, which is a huge impact for the patients. The patients can fall down and they lose their independence, but also non-motor symptoms that really drive the quality of life of these patients, the burden to the caregivers. And so examples of that can be sleep disturbances, where patients will suffer from insomnia, excessive daytime sleeping. They will suffer from apathy and depression, and cognitive impairment, all of which are critical unmet needs for the patients and opportunities for investors and companies like yourselves. So just to give you resources kind of at the Fox Foundation for these symptoms that maybe you're not so familiar with, we really try and systematically look at each of these symptoms and identify the gaps for these symptoms, and then we're working with communities, PD communities, the patients, regulators, also drug developers, to find out what the gaps are for these symptoms, and then how we can go in and fill those gaps. So I just wanted to re-emphasize the importance of symptomatic treatments. We're still not close to helping patients for these symptoms. And then kind of lessons learned. Sorry if I'm getting some echo. Hopefully that's a bit better. And then in terms of the lessons learned from the AD field, I think we're still a few steps behind the AD field in terms of biological acceptance of the targets. Obviously, the AD field has had their amyloid moment of linking a target to changes in pathology and then clinical measures. And then, as Brian mentioned at the foundation, we're really driving on many fronts across that whole pathway to enable that for Parkinson's disease. The other is robust biomarkers. The Alzheimer's field has a better biomarker toolkit than the PD field. They have their A-beta and tau, both molecular and imaging biomarkers. We're really trying to do that for Parkinson's disease with the acetylsynuclein assay that Brian mentioned earlier, but also really driving on PET ligands for alpha-synuclein as well. And then the third lesson that we should learn from the Alzheimer's field is around early intervention trials with at-risk patient groups. So patients that haven't got functional impairment yet, but they have biological indicators of the disease. And so at the foundation, we're building the infrastructure to carry out platform trials for these at-risk groups. So I would say those are the three areas that we really kind of can take learnings from the AD field. I'll pass it over to you, Stefan.
Well, thank you, Gaia, and I appreciate again you balance the need for disease-modifying therapies and the need for symptomologic treatments. From a drug developer's perspective, I think, first of all, what we've been seeing in the recent 3 to 5 years is a lot of excitement that has come back to the areas of Alzheimer's and Parkinson's disease to also address the disease-modifying, which is great to see. There's a lot of investment that has come back after companies had shied away approximately 10 to 15 years ago from that area because the pathophysiology, the blood-brain barrier, and other items have really not been well understood. And we are about to overcome that. And the number of earlier stage disease-modifying companies that we have on this panel, I think, is a great testament to that. As I said earlier, and I think this is important, this is why we at Merz believe there's not only a need, but there's also a continued room, even 20 years down the line, for symptomologic treatments, is that not all of these therapies will work in all of these targets. I mean, you addressed the possibility of having biomarkers, but it's also clear by saying biomarkers that you will be quite selective. And there will remain a pool of patients where the disease has either progressed already, or where these disease-modifying therapies don't work, where dyskinesias and movement disorders and depressions related to Parkinson's disease will need to be specifically addressed. And I'm very happy to report that companies like ours, provide a platform for partnering with biopharmas. And we have a number of discussions coming up today and the next few days during the JP Morgan week with companies who are playing in this space of non-disease modifying Parkinson's disease therapies, and we can provide that platform. I mean, and as a testament, I want to emphasize again, we have been inquisitive in that area. We've sort of acquired the assets of Accorda Therapeutics, and we believe this is a space that can meaningfully bring innovation to patients and improve significantly quality of life.
All right, great. So I agree. I think that symptomatic treatment is incredibly meaningful in this space. At the same time, and I think as we've acknowledged, there is disease-modifying potential represented from this panel. So the question about how the biology is translating into therapeutic targets and therapeutics themselves is one that I think has to be front and center for anyone who's invested in this space from a commercial perspective, from a unmet needs perspective. There are a number of different mechanisms, many of which are not brand new, have been incubated for a long time. Those targeting GK's metabolic pathways on the small molecule side, LRRK2. I don't think we have anyone on the panel representing alpha-synuclein, but that's kind of the analog potentially to amyloid therapies from the biologic side in Alzheimer's. And then, of course, cell therapy, dopaminergic replacement, and what that can be done through surgical implantation of dopamine cells into the nigrostriatum, and what that can accomplish for patients. So at the same time, none of these mechanisms are behaving in a vacuum. I think there's a belief that these things can be layered and stacked onto one another, so long as we're not talking about stacking toxicities. So maybe either Nick or Spring, if either of you want to jump in first, and then others, tell us a little bit about the modality landscape and how that's evolving in today's world.
Sure, I'll kick things off. So first, let's start with the fact that Parkinson's disease is very complex, multifaceted, and the patient population is very diverse. So just kind of talking about the symptoms piece earlier, the likelihood that there's going to be a silver bullet for Parkinson's disease, extremely unlikely. So we're going to 10 to 13% of the patient population is inherited. So approaches within that patient population are going to be different than an idiopathic approach. Cell therapy and approach tends to lend itself, at least initially, a non-edited forms of cell therapy for idiopathic. So for example, us from a disease modifying standpoint, we're a neuron cell replacement therapy that's been optimized for potency. And when it was developed preclinically, you have compelling preclinical models and FDA recognized preclinical models that shows our cells are highly potent. And we're hoping that that's going to translate into humans from a disease modifying standpoint. From a symptoms management standpoint, though, as well, levodopa, we're not taking our patients off levodopa. Maybe over time we'll be able to reduce levodopa. So symptoms management is probably going to be an underlying need for the foreseeable future, even with disease modifying therapies. Then again, within the disease modification, am I inherited? Do I have am I targeting a subset of that inherited patient population, like Gene's company or other companies, LRRK2, other enzyme deficiencies? So there's targets within the inherited and then the broader approach from an idiopathic standpoint as well.
Totally agree. On the disease modification side, I think I'm finally really excited about what's actually in the pipeline in the phase 3 with synuclein, with Roche taking the antibody in. I think for synuclein, we're going to get a pretty good idea. We have antibodies, we have small molecules, we have ASOs in the pipeline. We'll have a pretty good idea, I think, of that in the next 5 years, maybe much earlier than that. LRRK2, I'm super excited about. There's the LUMA trial that Biogen and Denali should be reporting on, hopefully at the end of this quarter. So all eyes on that. And then GBA, of course, and by the way, I tend to think of GBA, LRRK2, and then targets like ours, like USB30 or PINK1, all in the lysosomal mitochondrial axis. And we're seeing all of those now in the pipeline as well. So LRRK2, we'll see GBA, we're going to see there's the BL LTI molecule that should be reading out hopefully at the end of Q2. And then the mitophagy molecules are a bit further back. But I think we're going to see, and as was mentioned, I completely agree with this. I don't think it's going to necessarily be one molecule that's going to work for everybody. Even within that mitochondrial lysosome axis, you have GBAs like in the lysosomes. Would it be helpful if somebody has lysosomal issues already to put more mitochondria in there? Maybe not, although we haven't really fully tested that hypothesis. And the two communicated with each other all the time, so it's not as simple as one might think. Even with GBA, you see mitochondrial deficits and mitophagy deficits. And then likewise on the other side, if somebody has problems initiating mitophagy, even if you enhance the function of lysosomes, you may not necessarily be able to have disease-modifying effects. So I think, and I'm sure we'll talk about this, but I think biomarkers will be a really key component of which patients should get which therapies. But as far as therapies in the pipeline, I'm very excited about what's in the pipeline. And by the way, let me just give a shout out. I know you've heard their name like 10 times this morning already, but I think one of the reasons that the Parkinson's groups are so delightfully collaborative with each other is because of the Michael J. Fox Foundation pulling us all in and kind of almost forcing people to communicate with each other, which is fantastic.
I'm going to double click on the cell therapies. So just to follow on with what Spring and Nick mentioned, as we know, Parkinson's disease is a very complex disease with complex biology. So at Aspen, for our cell therapy approach, it's not just replacing the dopamine-producing neurons that are lost in disease, but actually a network of precursor cells. So both dopaminergic-producing neurons as well as supporting cell types that we hope and our testing now can actually help reestablish the neural circuitry that's lost in disease. And we think that will be an important benefit, not just for motor symptoms, but also looking at some of these non-motor aspects as well.
Yeah, I'll just pick up on that point too. So for us at Gain, what Spring had said, and it's backed up by what Sam said too, this complex biology, it is tackling one single substrate is probably necessary, but not sufficient to drive broader neuroprotection. When we look at glucose cerebrosidase biology, that particular enzyme has to move efficiently through multiple cellular compartments, and it has a job in every one of them. And as Spring has said, focusing on the lysosome might not be enough. Focusing on the mitochondria might not be enough. Taking the alpha-synuclein toxic substrate apart might not be enough, but maybe all three of those together, that might be sufficient to drive some neuroprotection, and then again give some cover to the brain to maybe enable itself to come up with some. There is a thing called brain plasticity, and if we can keep the storm in the brain down long enough, maybe there are some compensatory neural networks that can be developed, things like Sam is talking about. So it's important to target, I think, broad swaths of the biology, and that is disease modification. And it's going to take probably more than, I think we're naive if we think it's just one single drug that's going to cover it.
Yeah, and just to, hello? Yeah, okay. Just to re-emphasize, it's a really exciting time for the advanced targets. So that's LRRK2, GBA, alpha-synuclein. NLRP3 was another target that was mentioned earlier. It's a new inflammation target being pushed in the Alzheimer's field, but also multiple trials for Parkinson's disease patients that are ongoing. Again, watchfully waiting the outcome. So I think we're in a really good position for these advanced targets. The other area are new emerging targets for Parkinson's disease therapeutics. So at the initiative we have, at the foundation, we have a new initiative called Targets to Therapies that's looking at the next wave of emerging therapeutic targets for Parkinson's disease. Some of those targets we're effectively repurposing from other indications. TREM2 is a good example, OGA. So what data is there for those targets in other indications? What are the gaps that we need to fill to make them attractive for Parkinson's disease? And then also targets that are in early stages of therapeutic development. So there's molecules for those targets, but they've yet to move into the clinic. So what can we do to enhance the therapeutic development of those targets? Some examples are TRPML1, STING, that we're interested in, and then some very novel targets where there's a good package of evidence for those targets for Parkinson's disease, but very limited therapeutic development. All to say that foundation is really thinking carefully about the next wave of emerging therapeutic targets, making sure there's kind of the second wave of targets for patients.
Excellent. All right, another topic that we covered in our prep session was around endpoints and biomarkers. And just thinking beyond the UPDRS, there's a lot of innovative, novel endpoints, including digital and wearable endpoints. I think Roche came up with an iPhone-based endpoint for the prasinezumab study. And these can be important for things that Gaia mentioned earlier, like gait and sleep disturbances. And then also thinking about what is the trajectory of those endpoints to define disease modification versus symptomatic relief. So there's a lot of different concepts in there, but I'll ask our panelists to weigh in. I don't know, Gaia or Spring, if you want to comment.
At the foundation, we are definitely very interested in digital healthcare and wearables for the patients. We're funding groups across many clinical endpoints, including gait and mobility, but also speech and sleep wearables. We're also very closely working with regulators around how to get these tech consumer devices into the hands of patients as well.
Yeah, I just will add to that. I think the wearables just lend themselves so well to an indication like Parkinson's disease, both from motor and also things like sleep. So I'm actually kind of surprised we already don't have them approved, and hopefully that will change in the near future. But then I think on top of that, we also need biological kind of readouts. So the synuclein, for example, seed assay that the Fox Foundation has developed is an excellent starting point. I hope that this is the first domino of many biomarkers that we can look at, including, for example, for mitochondrial function, for lysosomal function, and things that may, the seeding assay is great right now, but it's difficult for progression. It's great for diagnosis, I think. So if we can continue to add to that, and if we can learn, again, going back to the Alzheimer's, they have tau and amyloid and NFL, and it would be really nice to just have more of those. And I know that there's more investment being made in biomarkers, which is fantastic too. So hopefully that will improve. But I think that's an area that we can have more, and it would be really, really informative to the field.
Yeah, just to add on to that, so at the Fox Foundation, we're funding groups to make this synuclein seed assay more quantitative, so it can be used in longitudinal clinical trials. We're also trying to move the assay, at present it's in the CSF, to a skin-based assay. So just echoing just the utility of this assay for clinical trials, particularly early risk platform trials, as I was describing earlier. We're also spending significant resources around additional molecular biomarkers outside of alpha-synuclein across all the different pathways involved. One example is our neuroimmune consortium that's specifically designed to assess neuroimmune biomarkers using our PPMI samples and standardizing assays and sample collection from patients.
Just back to the wearables piece again. We're cell therapy, so we're going to take a look at imaging. We're going to look at engraftment and innervation. We're also going to do the classic movement disorders, neurologist assessments, MDS, UPDRS, part 3. But what's really important is how the patient feels at the end of the day. And wearables, I think, when you're looking at everything from sleep to gait, are probably the best translation to facilitate the patient's interpretation of how they really feel, the feeling improvement. So it's a good marker.
I want to pick up on that point a little bit because, at Gain, when we started our phase 1B study in March of last year, in 2025, we took a very, very hard look at including a wearable technology there. And I think one of the challenges that we all just need to be aware of is that technology has to be very, very simple. It has to be elegant, and it has to be very easy for somebody or for patients that really can't think about that sort of thing and the technical support that they might need. That might be one of the reasons why it's been a little bit slow moving in order to integrate these more into earlier stage studies. But I think the digital technology is coming along. I know that we're constantly reviewing all this technology at every single conference because it does seem to make another step. At every single conference, it gets a little bit better. The progress is being made. So we're excited about that, and hopefully introducing something in our phase 2 study, which will open up that door to more potentially objective clinical reporting. The UPDRS score is great, it's the gold standard, but it's just so prone to subjectivity that it gets tough. It's a tough clinical marker. The digital technology offers us a way around it. It's coming quickly, and I think it will start to be in studies beginning this year, hopefully ours, and it'll be adopted more and more.
I'd like to build on that point of digital technology and wearables to be really practical. And I'm thinking, of course, about this beyond clinical trials. I think it is important for clinical trials if it's well aligned with the regulators. But beyond that, we've had a look at many, many different wearable and digital solutions, and they need to be practical, and they do need to be pragmatic for the patient to use. And this is where currently, all of these technologies that we have had a look at, and we've assessed over 10, probably 15 different projects with companies, they've all failed, unfortunately. Progress is made. The breakthrough, I think, has yet to come. And that is across sort of gating problems, movement disorders, essential tremor, and other things that we've had a look. I hope the breakthrough will come, but it is yet to be seen. The first step is always the clinical trial, but then life happens beyond the clinical trial. And I think this is where sort of the rubber hits the road, and we've not seen quite the progress that we'd hoped for yet.
Just one thing to add, and I'm probably going to tee it off to Gaia. There are these great technologies that do evaluate some of these non-motor aspects and quality of life components. And I think one of the questions is, what is clinically meaningful, and how does that translate to regulatory approval or regulatory support? So if you see improvement in gait, in freezing of gait, fall risk, in sleep, what does that actually mean? How much is good enough? How do you engage with regulators to have a meaningful conversation from a quality of life perspective? And what are the standards or level of benefit you need to see on some of those other endpoints? I think that's something the Michael J. Fox Foundation can help inform and engage on.
Yeah, and then the final class of kind of endpoints to think about is our imaging endpoints. Yeah, okay, great. Yeah, it was Jean's fault. The other class are imaging endpoints. So we have the DAT scan, and we're funding groups to help improve imaging quality and then the analysis of those DAT scans, so we can reduce the number of patients that are needed to be enrolled into clinical trials. And then the other big improvement, I would say, in the last year has been the alpha-synuclein PET scans. We're funding multiple groups that are carrying out work here, and we've seen really exciting new results in the last 12 months. So that's another space that will be useful to watch in the next year.
All right. Well, while we're talking about all these different things that can help expedite and make trial design more efficient, obviously, with Parkinson's, it's a neurodegenerative disease. These are long trials. They're expensive to conduct, and often with high risk. So where are we in terms of historical use of incorporation of historical control, synthetic control arms, I guess other things that could potentially help de-risk these studies and get to an answer sooner or more cheaply? And where are regulators in terms of accepting those? Maybe I'll ask either Sam or Jean to weigh in on that.
So I'll go first. So placebo arms in Parkinson's, it's a tough one. That's an obstacle because these trials, especially as they get to registration stage, are long trials. They're 1-2 years, and to keep a Parkinson's patient on placebo for 1 or 2 years just doesn't sound right. Doesn't seem right. But there are other creative ways to design trials. I know the Michael J. Fox and other groups are thinking about portfolio studies where a group of sponsors essentially shares a placebo group. That can help preserve a lot of patients who are randomized to something that's probably not active. That comes with other challenges too. Sponsors are very, very different. They have very different complex issues. Protocols change over time. Sharing a placebo group can work if in a very, very tightly controlled environment. Surprise, where the regulatory interactions are organized very, very tightly. I mean, a lot of portfolio trials, I think, have a little bit of a way to go before they are something that we rely on heavily. But trying to deal with offering something to patients potentially in terms of a crossover design, things like that. Those are all things that we can start to think about if we start seeing efficacy in some of these disease-modifying drugs, such as GT-02287, which we're developing, and be able to get a time point, find out when we're seeing clinical efficacy, and then get as many patients crossed over as soon as possible.
Yeah, so I mean, there are case-by-case examples of regulators showing flexibility, not specific to Parkinson's, but mainly with rare diseases, using real-world evidence or other innovative tools to have faster trial designs. Obviously, for us, Not rare diseases, but ethical components certainly in developing medicines for patients living with Parkinson's and older disease populations. So it feels like a really wonderful moment for industry and regulators to collaborate closely on how to have shorter or smaller placebo-sparing studies while still maintaining scientific rigor, especially because PD does have, a historically large placebo impact. Some of these tools include, AI-enabled tools or potentially expanding longitudinal data sets to support faster, more ethical trials. And I think in order to do that, certainly the voice of the patient is very key. And patient advocacy groups working with regulators, working with sponsors, working with teams and patients and caregivers to help drive toward some of these goals.
Yeah, just building on the ethical point, Sam and I have interventional therapies. So think about a placebo control arm with an interventional therapy. So the solutions that Sam offered, AI generated, comparing to natural history, you can tell the FDA wants to move in this direction. They just haven't moved in this direction yet. And then the further complexity is, along with what Guy also said, is not just what the placebo design looks like, but again, ethically, how long do we make these patients wait before they can opt back into therapy as well that are on the placebo arm? So it's complex, but FDA is signaling, but we're not there yet, whether different alternatives are possible.
Yeah, at the foundation, we're setting up an initiative. It's already been launched called Pathway to Prevention, where we're designing and building the infrastructure for a platform trial for Parkinson's disease, where you can effectively test multiple molecules or therapeutics in parallel, where the control arm can be shared. This has been carried out in other indications like Alzheimer's disease and ALS. So you effectively share the costs of the design of the trial, and also it can be adaptive, so you can have additional drugs added to the trial during the course of the trial. And then at the Fox Foundation, we're focused on a platform trial where we have early patients, so early at-risk patients, so they have evidence of the biology of the disease, like the seeding synuclein amplification assay and the DAT scan, but yet they haven't got functional impairment. So that's another piece that we're kind of lessons learned from that Alzheimer's field. And really, the idea of a platform trial kind of in the short term is to get buy-in from all the stakeholders who are involved in that trial. So the patients who need to be enrolled into the trial, pharma who will provide the drugs, regulators so they can get confidence in the enrollment strategies, and then also the endpoints and outcomes. And then ultimately, the idea is then to provide confidence to folks like you in the room, where you can then run your own independent early at-risk trial for Parkinson's disease patients. And then in terms of the PD field, the time is kind of right at the moment for starting these early intervention platform trials for Parkinson's disease patients. Through our biomarkers initiative, we've got these deep phenotype longitudinal cohorts of individuals. Secondly, the regulators are coming on board. We have this new biology staging framework for Parkinson's disease that's been accepted by the FDA. They actually co-authored the paper on the staging framework that we want to use. And then folks like you in the room are coming around. We've got a robust, growing pipeline for Parkinson's disease where we can move some of these therapeutics into a platform design trial.
Excellent. Thank you for those comments. And we also wanted to think beyond Parkinson's disease to other synucleinopathies like Lewy body dementia, and even other movement disorders like essential tremor. We saw some positive clinical trial results from Praxis in essential tremor last year, and tardive dyskinesia, Neurocrine and Teva have made a lot of progress there. Are there other synucleinopathies or other movement disorders where we can leverage some of the lessons learned there to Parkinson's disease? I don't know, Stefan or Nick, if you want to comment on that.
Yeah, very happy to. And I think these movement disorders that you mentioned, essential tremor, tardive dyskinesia, they're all very different from a disease pathophysiological pathway. So this is also why we've seen in the past quite a number of failures, actually. There's been some progress recently, but I think the big breakthrough still needs to happen. We are also seeing, when it comes to levodopa-induced dyskinesias to LID, we are seeing some interesting sort of progress that's being made in the earlier phases of clinical trials. We again here we believe there is a chance for being very specific to the specific patient, trying to identify biomarkers and the underlying disease pathophysiology in order to develop sort of targeted diseases. And I think the proof is still in the pudding if that is going to hit the market. So we are watching that space very carefully, and there are some select advances that we are monitoring carefully, and we believe could come to market next 3 to 5 years.
Yeah, scientifically, there's very little overlap between these movement disorders and Parkinson's disease. Probably the only thing might be the connections, the motor skills connections in the brain as an overlap as a source of dysfunction. But other than that, the original cause of the disease, it's all completely different. I mean, tardive dyskinesia is a result of a side effect of other drugs. It's created by the use of depression drugs, et cetera, that block dopamine. So I think, but that said, from a research standpoint where movement disorders neurologists are involved, assessments, there might be some commonality and shared learnings across the space from an assessment standpoint that are possible. But primarily, there's a big difference. Even commercially, these are big. Parkinson's market's about 7 billion TDs. There's 2 products that are about 2 billion each. ET is small in device, and dystonia, my friend down at the end of the panel, him and AbbVie own that space with tremendous success. So it's different commercially, it's different mechanistically, but there might be some possibility on the movement disorders assessment side that we might learn from each other, I think.
Can I add also, this is not a disease per se, at least not recognized, but aging itself is the biggest contributor to all of these indications that we're talking about. And I think, I used to think of it as like, okay, aging is the biggest risk factor. But if we think about it as the biggest contributor, hopefully that's a mind shift, and it's a contributor that can be modified. So we tend to think of aging as something that's just happening in the background and is unmodifiable. But now we're seeing that, in animals, you can just flip one gene and the animal will live twice as long. And you see a lot of the same biological pathways involved, mitochondria, immune, which was discussed earlier. All of these are components of aging. And if we can, learn from the aging field and vice versa, aging field is surprisingly young, actually. But I think we can learn a lot from that and consider aging as a contributing component that is modifiable itself. That will help us with Parkinson's disease as well.
All right, I think we have a few more minutes. Maybe I'll throw my panelists a little bit of a curveball. And if there are any questions from the audience, please keep them in mind. But so, Stefan, I know that you're fielding potential in-licensing pitches. Many of the companies represented in this room are going to rely on non-dilutive funding on a former partner. What are some things that stand out for you when you're looking at pitch decks? What are some red flags? Is it all about data? Is it kind of more about the patient journey and the ability to bend the curve with patients? Like, what are you guys looking for? And then maybe others can speak from a perspective of being on the sell side of the pitch.
Yeah, great question. I think data, of course, is capital. When we look at any VD opportunity, understanding the disease, understanding how the drug delivers on the disease is, the key thing. And then the data, early, mid-clinical data should speak for themselves. The second piece is an aligned pathway with the regulators, ideally with a solid biomarker that is aligned with the regulators as well. That is important. And then the last piece, and I wouldn't underestimate that, is actually a fit from a patient pathway as well. I mean, we have a very, very specific target, how we treat sort of movement disorders, how we treat symptomologic, as I said, how we treat symptoms, and it just needs to fit our commercial and go-to-market model as well. And there is a cultural fit as well for any of the targets that we look at. That is very important as well. I mean, people really understanding the disease well, taking it serious, and having a long investment in that is something that we have and we look for in partner companies as well.
This morning's BD panel, the big pharma BD panel, talked about the importance of preclinical data, raw preclinical data, before they're going to, in addition to what Stefan said, before they're going to, and I can speak that even from a financing standpoint or a partnering standpoint. We raised $82 million on the back of our preclinical data, showing superior potency preclinically side by side with the other cell therapy providers in the space. So tight preclinical package, raw data, and to Stefan's standpoint, strategic fit from a partnering standpoint will matter a lot.
Yeah, those are great points. Just to add and echo, of course, good data always helps and is critical to achieving financing and partnering goals. Aspen also raised $115 million Series C and included a strategic in that financing. So in addition to the data, I think once you start generating the clinical data and move beyond preclinical, a lot of the questions we were getting is the cost to actually get this to market and the cost to then increase sort of the opportunity and market share post-launch. So sort of like once you got the data, it's like on to the next set of things you need to answer questions about. So being able to, cover multiple aspects of how to actually operationalize through development and beyond is, I think, also really important.
I can approach it from the other end, which is from the sort of, we're one of the only ones, I believe, that are publicly traded, small molecule developing publicly traded Parkinson's companies. And it's been tough with investors. There's a lot of biology here that's not well trodden, and we see a lot of gaps, knowledge gaps, from investors in terms of the understanding. And that's our burden to bear. As Gain moved through its early clinical studies, well, we're still early, but as we moved through our first in human, we heard target engagement was going to be very, very important. We had to show that GT02287 was modulating glucocerebrosidase. Were we on target? And we were able to demonstrate that in a couple of different iterations of healthy volunteer studies. And then, okay, great. So GCase, you're showing that it's moving around the cell a bit better and more effectively. What is that going to mean? So in phase 1B, where we enroll actual Parkinson's patients, can we characterize that disease cascade a little bit better? So if you go to, and I hate to blow them up, but if you go to Jonas's talk at 240, we have started to unpeel the cascade we're targeting and further characterize it better. We have to characterize these pathways better. Are you getting to GCase? Is that affecting the cell? Are you seeing improvement in the mitochondria? Are you seeing improvement in the lysosome? Are you seeing other toxic lipid substrates correlate with a healthier cell? And again, go to Jonas's talk, because we've only had data in-house for about 2 weeks, but we're seeing some striking correlations in some of the biomarkers that we think are now becoming very important. But along the way, it's been show me this, show me this, to get to the next financing, to be higher up on that list of companies that pharma calls when the press release comes out. Hey, where are you going to be? When can we catch up again? It's been a road, and we'll continue to walk down that road. We're going to get a phase 2 this year. We'll either finance at our own or we will partner based on the work that we're doing now. But it has been a show me story the whole way out. There has been very little faith extended without data. So good data is always gold. We at Gain have been what becomes clear to us is that we need to characterize this particular, how we're intervening in the disease cascade very, very well. And it has to tie to something in the UPDRS score. And we think we're there. And we'll probably get more visibility on that through this year. I didn't mean to plug the company or anything, but we're talking about biomarkers, and we've been sitting on this data for 2 weeks and mulling it over. And I think it's important to talk about how we're constantly trying to advance the narrative and characterize these things and inform the field. Michael J. Fox, thank God, is around, because for all of us sponsors, we kind of want to keep we want to play our cards close to the chest. And where do we go to see if anybody else is seeing something similar? Is this something that's interesting, or how can we get this prioritized by you folks, because you have way more people and way more resources. And Michael J. Fox has been fantastic at that the whole way through. There are other disease areas that have that kind of thing, but we're lucky in Parkinson's to have Michael J. Fox.
Yeah.
So I'll end on that.
Great. Okay, so I think we're gonna finish it up here. I think just to recap, the biology is critical. We're seeing a lot of translation of targets that have been around and modalities that have been around. It takes a long time, but I think we're bending the curve. I think we're turning a corner. Jay and I have the privilege of doing this every year, so I think we get a longitudinal view, and things seem pretty positive. It's not just about the science, though. It's also about the trial design. It's about the endpoints. It's about the patients, incorporating the patient voice, working with regulators, working with the community, thinking beyond Parkinson's to areas of adjacency. So we covered a lot of ground in an hour, and I think it's been really a testament to our panelists here today. So thanks again for your attention, and we'll stick around if there are any questions afterwards. Thanks, everyone.